Okay, good morning to all of you. So in last lecture, number 11, we have seen about the cytology as a source of data for the systematics. So next our fifth point is there, fourth point, embryology as a source of data in the systematic. So basically, while considering the embryology, so what is the embryology? So this data, generally, there is a one authority is there, or there particularly in the second half of the 19th century. Generally, it is which is utilized in the 20th century. So many embryologists, like our Indian Professor Maheshwari in 1930, then Kapil in 1962, then Johri in 1963, and Davis in 1966. So they have given the idea about the embryology. So what is embryology? So embryology is the study of sporogenesis, gametogenesis, fertilization, embryogenesis, then embryo development, post-fertilization, endosperm development, and the seed development. So generally, while in anatomy you see the specific words are there. So like this in embryology, these words you have to remember properly, right? Their spellings and their references. So what is sporogenesis? Sporogenesis is related to the pollen grain or the spore development. Gametogenesis means gametes development. So in angiosperm, gametes are developed in the suppose the male gamete is nothing but the pollen grain. After germination, it develops the pollen tube. So there is the male gamete formation, and in ovule there is the development of egg cell. Then there is a fertilization, fusion of these two gametes. And after fusion, it forms a embryo, or firstly zygote to embryo, and this process is called as embryogenesis, embryo development. After divisions, redivision, or the post-fertilization is there. We will see the process. So here the or in the next semester we have in this separate paper embryology. So that time all developments are there. So it gives to the formation of endosperm and the itself to the formation of a seed development. So all this it is called as the embryological study. Then what is microsporogenesis? So as there is a pollen mother cell is there, commonly it is called as PMC, and pollen grain development, it is called as the microsporogenesis. Then megasporogenesis includes the nature of archisporium in the ovule. Then embryo sac development, fruit development, endosperm development, and the embryo development in the seed. So you remember the concept or the foundation meaning of these two words. Then as here you are observing some diagrams are there. So it is nothing but the how the embryo development takes place, how it becomes a multicellular structure is there. Just we are observing the seed, fruit, or like this, but it having very specific development is there. Nextly, these are the some steps are there. So just see how the pollen grain develops. So microspore mother cell, microspore become 4 nucleated or 4 celled. Then it shows the fusion is there. From this tube there is a fusion, it forms 2 cells. Then germinating pollen grain, and it forms the 4 gametes are there. These are nothing but the sperms. Same thing at the time in flower, carpel is there. So it gives the megaspore development in the ovule. This megaspore, after that, it gives the formation of 8 nucleated embryo sac. You are observing these green diagrams are there lower side. So embryo sac is there, and again there is the fusion with that of the this one. Then what is post fertilization? Again there are some changes. Zygote, embryo formation, then seed formation is there, and again we are observing it is seen in the all these changes takes place into the flower itself. So therefore, diagrammatically, we can understand this embryological study. Then nextly, the presence and types of the anthers. Generally, these are different in the different, particularly in the angiosperm. So we are knowing this is stamen, and it shows the anther. So anther, when we are taking the cross section of this anther, so it shows the tapetum, outermost layer protecting. Then vascular supply is also there. Endothecium, that is inner wall, is there, and the microspore mother cells region. So it is called as the tetrasporangium. The notch is present to this anther. It is called as the stomium. So it is generally for our understanding. Then nextly, development and organization of pollen grain is there. So just I have selected randomly. So on this study, it is as a source of data in the study of angiosperm, because each species, each genera and species having the different character. So here you are observing development. So how is the pollen grain? These are the scanning electron microscopic photographs, and in this one you are observing here. So you just see how many layers are there. So here there are they show the magnified. So outermost layers, and these are shown by the scientist Earded Man, right? So specifically endospore, exospore layers are there. Again they are divided into 1 to 3 layers in detail, but these are very microscopic. Without scanning electron microscope we cannot study in detail. Then different types of the microspores are there. So there are these main 4 types: tetrahedral, isobilateral, decussate, and T-shaped and linear. So this tetrahedral means 4. Then isobilateral means 2 upper side, 2 lower side. So iso means same, bilateral means on both sides, right? Then decussate means one pair is opposite to the other, diagram C. Then T-shaped, English letter T. So you see this arrangement, it is not seen in all, but it is seen in some example. We are not for your special study, because we cannot give them more weightage, but just you remember the outline of these microspore tetrads. So after division we are, last one is the linear. Linear means all spores, they are in the one line or one level. So therefore it is called as the linear. Then nextly, these are some examples are there. Trapha, that is we are knowing. It shows the Onagrici family and monotypic type of the family. So based on this photograph, so it is the aquatic plant is there, like our typha. पानकणीस माहिती आहे त्यासारखाच हा पाण्यामध्ये असतो. And secondly, it having the other fruit. Generally it shows the drupe type of the fruit is there. So fruit character it is also under the embryological study. The number and arrangement of the anther locules are there. Just we have seen above. So anther, filament, the TS of this anther is there, and tetrasporangiate structure is there. This is the tetrasporangiate structure. Already we have seen in the above. Then these diagrams, they shows the steps of the anthers. In the anther, how the pollen grains are developing. So just I have selected this for our interest. So how the anther, when it increases in the shape and the size, so how generally it shows the character. So therefore we have to consider this, and it developed to the formation of tetrasporangia. Mature pollen grain, it is developing inside to this. Then nextly, these are the some germination of the pollen grain on the stigma of the carpel is there. So we have discussed how gametes formation are there. So it is also typical, because the pollen grain of the, suppose, hibiscus. So it is germinating only on the hibiscus, or pollen grain of the your sorghum or jawar or wheat are there. So they are chemically identified by the female plant, and therefore there is a process of fertilization is there. So just you see A, B, C, D diagram, then pollen tube formation, then there are the male, there is a tube nucleus is there, and it shows the development of male gametes are there in this pollen tube. So it is a good process generally seen in the all angiosperms. Then so these two processes, one is called as a microsporogenesis, and here the gamete formation is there, it is called as the microgametogenesis. So what is microsporogenesis? So development of the haploid pollen grains. And what is gametogenesis? Development of the gametes in the pollen, right? Pollen tube formation and the gametes formation. So please remember the differences. Then second part is there our in embryology. So in embryology study, the development and structure of male and female reproductive organ, it is important. Here you are observing our flower structure. So generally we are discussing flower. It is hypogynous flower, epigynous flower, perigynous flower, right? So what is this? So diagrammatically, as the development of this floral arrangement, either it is below the ovary, then it is called as hypogynous. When it is above the ovary, then it is called as the epigynous. You see the last diagram D. And when it is in between the ovary, then it is called as the perigynous, right? So therefore this development is important, and diagrammatically this representation development of embryo. So how embryo develops? So integuments are there, endosperm is there, and the nucleus is there. Then developmental structure after this, the ovules are there. So what are the ovules? So structure and development and position and the types of ovules in angiosperm. So mainly in embryology as a source, there are different examples are there. So first type is there when A, you see the A diagram. So it is called as the micropyle, that is opening of ovule, and the basal attachment, it is called as a funiculus. So you see A diagram. So they are in the one line orthotropous. B diagram, micropyle and funiculus on the same side. It is called as the anatropous ovule. Then in the C diagram, it shows the compylotropous in the 90 degree. Then D diagram, it shows the ambitropous. It is 90 degree, but shows the slight, there is a fluctuation is there. Then in the E diagram, as it shows anatropous, not totally, but some angle is there. And F diagram, it shows the cercinotropous, so it shows the total coiling is there. So particularly I think it is seen in family Cactaceae for this one, Opuntia किंवा निवडून वगैरे बघतो आपण त्याच्यात हा शेवटचा टाईप पाहायला मिळतो. त्याचे एक्झाम्पल्स वगैरे दीज आर इंटरेस्टिंग, पण जस्ट our this topic you cannot give the more weightage here, but just I am giving the some examples here. So many examples are there. Then you remember these types: anatropous, orthotropous, compylotropous, ambitropous, right? Then types and development of embryo is there. What is embryo? Already we have seen. So this, it shows the two ends: upper end chalagen end, lower end upper micropyle and lower chalagen. So antipodal 3 cells are there, 3 polar nuclei, then central cell is there, egg cell, then 2 synergids, and there are the Philippon apparatus is there, right? So monosporic embryo sac, bisporic, and the tetrasporic. So based on this embryo sac nucleate condition, it is commonly divided into monosporic, bisporic, and the tetrasporic. Then you see this diagram is related with that of the detailed structure of this embryo sac in there. Diagram is the same. It is also, you see, it is the 8 nucleated, above 3, basal 3, synergids and the egg cell, and antipolar cells are the 3, and polar nuclei are the 2. Then this our standard diagram is there. How the ovule, ovary, and these are there. Arrangement of these ovules are there. So here second male gamete N fusing with that of this female one. So after at the time of germination, it entered to the micropyle, and there is a fertilization process is there, and it is called as the syngamy. Then after that there is a porogamy is there, or chalazogamy and mesogamy, types of fertilization. Porogamy means from the one pore, chalazogamy means from the chalazal end, and mesogamy means it shows the end. But their examples are the different. So male gametes are haploid, female gametes are haploid, then synergids are there, polar nuclei. So they use the formation of the either zygote then endosperm. After fertilization, it shows the endosperm development. So what is there are 3 types of endosperm: nuclear, nuclear endosperm, cellular endosperm, and the fallopian. So you see, nuclear means in the nucleus generally, cellular means the many cells are there, and fallopian means at the margin. So they show these main 3 types. Then growth of colon tube is there. Already we have discussed this, but I have selected as due to the diagram is there. Then nextly, there is the what is meaning of Microsporogenesis. We have seen microsporogenesis. Micro means what? Colon then develop. In Microsporogenesis, there is the formation of a, it is commonly seen in the ovule. Right now, embryo is there, and so here you are observing the steps having the development of the megaspore. So how megaspore develops? So megasporogenesis and the megagametogenesis, gametes form. So how this develop into the gametes, right? So archegonium we are knowing in algae, fungi, bryophyta, pteridophyta, antheridium, and archegonium. So in archegonium, these words are we have seen, but here in angiosperm the conditions are different. After that, the ovule arrangement is there, and it develops the ovary, TS of ovary. So in practical, we are taking the section TS of ovary. So marginal placentation, then in dicot and monocot, there are different placentation, marginal, then bilocular, trilocular, tetralocular. You see diagram C1, C2, C3, C4, right? Then B2 diagram, it is the parietal placentation in cucumber like this one, or the others are there, precentral placentation in tomato or like this, diagram E. So photograph E, it shows the precentral. In angiosperm, the monocotyledons and dicotyledons we are doing. So single cotyledon, these are grouped under monocots, and 2 cotyledons grouped under the dicot, right? So at the time, what technically we may say, which curry today we have yet, either monocot or the dicot. So based on this character, then especially family order Orchidales is there. So the types of in Orchidales, the undifferentiated embryo is seen, and it is very little or there is no endosperm, Orchidaceae. In the other fruits are there, nature of seed coat, it is different. For example, apple is there, or pear fruit. So it shows different types, and it is called as the parthenogenesis. The monocot versus dicot seeds are there, so they are having the specific character. Epicotyl, hypocotyl. Monocot seeds are nothing but the maize or sorghum or the bajra are there, wheat is there. And in dicot, your dicot members are हरभरा असेल किंवा डाळी म्हणतो आपण त्याला, सो they comes under the dicotyledon. So therefore, embryology is used to solve many problems at family and general level. For example, family Monograceae, embryo sac is 4-nucleated with microbial megaspore dated and not in the other family. Trappa is the another example, family Onagraceae. It is aquatic, solar petiole, 2 types of leaves and semi-epinynous ovary. Thirdly, the family Podostamaceae is there. It having unique feature and it shows the embryo sac due to the disintegration of micellar tissue, pollen grain impairs bitechnic ovule and the bisporic embryo sac. So only 2 spores are there in embryo sac. Generally there must be 8, but here the 2 spores are there, gametes. Then family Loranthaceae. It is a reference given by our Indian scientist Maheshwari, 1964, based on characters divided into 2 subfamilies, Loranthidae and the Discoidae. So what is Loranthidae? Pollen grains with tri-radiate mark, polygonal type of embryo sac, that is 8-nucleated, and early embryogenesis shows biserial. Example Loranthus. आपल्या त्याला आपण पॅरासायटिक प्लॅन्ट म्हणतो बघा, लोरॅन्थस. सो इट इज कॉमनली ग्रोइंग ऑन द लार्ज ट्रीज. देन डिस्कॉइड मीन्स वॉट? हिस्कम. पोलर ग्रेन स्पेरिकल एलियम टाइप एम्ब्रियो सॅक, म्हणजे 8-nucleated but 7 spores are there and early embryogenic. So therefore this shows the biserial. Example Hiscum is there. So you remember this good reference for our study. Then nextly, fourth example in family Cyperaceae. Cyperaceae म्हणजे मराठीत आपण त्याला नागरमोथा किंवा लवाळा म्हणतो, त्याचे मेंबर्स. सो फॅमिलीचे. सो कॅरेक्टरिस्टिक हॅज only 1 pollen grain form the microspore mother cell by meiosis and 3 are degenerating and 1 is the functional. According to Shah 1962, all genera and species shows presence of single functional microspore. या फॅमिलीचे सगळे मेंबर्स सायपरस ऑदर्स आर देअर. सो दे शोज दिस कॅरेक्टर. The 5th number characters of angiosperm divided in dicot and monocotyledons based on embryo. So this is the important. And 6th point is there, based on embryological features, order Polymonians and Plumbaginians related to order Caryophyllens. Actually these are the concluding points are there, but embryological study it is considered for the classification of the families also. Okay, so this is our today's lecture. In the next lecture we will see the detailed part.